Hello, my name is George Scheibel, and today I'm going to talk about biological complexity, specifically the cellular differentiation within multicellular magnetotactic bacteria and the implications to the evolution of complex life on Earth. To begin, I would like to introduce the idea of complexity in regards to the evolution of life on Earth using a simple Cartesian coordinate system. For the first cell to evolve, simple molecules had to interact to form more complex molecules that eventually yielded the first cell. There are many great talks at AppGradCon that discuss this first step in the evolution of life, but I'm going to focus on the next step, which was when that simple cell continued to evolve into a multicellular organism that eventually gave rise to life that we see on Earth today. Of course, at certain steps in this evolution of life in regards to complexity, some cells plateaued and remain the same, giving the diversity we see on Earth today. It helps to have criteria for multicellularity to describe what we observe as far as multicellularity in organisms. The first criteria being that the organism is built from several cells of the same species, has a specific shape and organization, as well as synchronized growth, so this would exclude things like cancer. There would be no competition between cells, and they would exhibit a coordinated behavior and response to internal and external stimuli. And they would do this using cell-to-cell signaling. And finally, there would be an existence of a division of labor. Something like the cells in our lungs and stomach are from the same organism, but they're performing a unique and different function for the entire organism. The organism that I study is multicellular magnetotactic bacteria, or MMB for short. And this organism is multicellular in that it's composed of several cells from one species. It's magnetotactic, which means it's capable of sensing Earth's geomagnetic poles that it uses for navigation. And it's a bacteria belonging to the Desulfobacteriota. In this SEM image on the upper left, I show two MMB that have potentially just undergone division, and you see that it's a small consortia of cells that are tightly packed together, forming this ball-shaped structure. In the backscatter electron image in the lower left, you'll see it's MMB, and you'll see these white lines within each cells. And these are the magnetosomes, bacterial organelle responsible for magnetotaxis of the organism. The TEM is showing a slice of the MMB where you see individual cells organized around an acellular center, in which you see these black dots in this cell in the upper left, and these are the same as the white dots you see in this backscatter electron image, the magnetosomes. And these are synthesized by the MMB. It's a mineral called greigite that has a paramagnetic dipole. And they form them in these chains that essentially act as a compass needle, allowing them to sense Earth's geomagnetic poles that they use for a taxis in the water column to find their desired redox state for survival. This organism was discovered in 1983, and we still have not yet been able to bring it into culture, which complicates all the studies, so you can't simply throw genetics at it to test really interesting questions because we cannot culture them yet. So all the techniques used in this study are culture-independent techniques. The MMB are an obligate multicellular organism that have a putative life cycle shown here in this panel SEMs, where the organism grows in size and then eventually divides. And similar observations have been made using microscopy. MMB cannot survive as single cells either. And here in this lower left panel, you see a live dead stain where cells that are stained red have lost their membrane integrity and have died. And so here you see two dislodged cells that die, and eventually this loss of cells compromises the integrity of the entire consortium, and the whole consortium dies. In the SEM panels on the right, we look at A, a healthy MMB, and then B, C, and D, some different scenarios where cells have become dislodged and the organism, the entire consortia has collapsed. The MMB I study are found at Little Sipawissa Salt Marsh, located on Cape Cod in Massachusetts. And here I show an aerial view of what the site looks like. And it's a brackish site, so there's an influx of seawater from the bay there, and then fresh water coming out towards the bay. Here's a ground level view of what the site looks like. It's just a small marshy pool. And the bacteria that I study are just found in the sediment of this pool. Now, the video I'm going to show you is the MMB are in the bottom of this Eppendorf tube, and they're swimming up towards this magnet that you see here. You see them slowly taxiing in this 20-minute time-lapse video. In the next video, it's a hanging water droplet with the MMB swimming towards the edge of this droplet, where I show a cartoon of the magnet that is present there. I then flip this magnet, and the individual MMB all start swimming in the opposite direction, now thinking that the magnetic north is in the opposite direction. Just so to note, each little dot you see here is a single MMB consortium that's swimming. Now, next I switch the magnet again, and you see the MMB switch their swimming direction, moving back towards the edge of this water droplet. Now, when we return to the criteria for multicellularity in regards to MMB, we see that several criteria are actually met, in that they're built from several cells of the same species, they have a specific shape and organization, there's no observed competition between cells, and they exhibit a coordinated response, as we see by an external stimuli of a magnetic field that coordinates their swimming. We still are investigating cell-to-cell signaling that we're using genomics for, and we want to know more about a division of labor within MMB, where we may potentially observe differences between the function of cells within the consortia, shown here by using different colors. So to begin, we want to do whole genome sequencing. So we sampled sediment from the marsh and stirred that sediment up and put a magnet next to it for about 60 minutes, and the MMB will swim up out of the sediment towards the magnet and form this small cell pellet you can see here that we can then simply just remove by pipetting. We then took that sample of MMB and sorted them using fluorescent activated cell sorting to isolate the population of MMB. And we sorted them into a 96-well plate, where we did multiple displacement amplification and short read Illumina sequencing to get 22 MMB genomes. We then used the MMB genomes to address whether or not the consortia is clonal, in that is the genome in one cell the same as the genome in its neighboring cell, addressing this division of labor question. And what we found were they were not entirely clonal. So what this sequencing project did is we generated several libraries from a single MMB in a well that was sequenced, and we mapped those libraries back to the best assembly to identify single nucleotide polymorphism differences within a single MMB consortia. And this work done by Frederick Schultz at JGI found that MMB have a higher rate of variance, SNPs per genome, as compared to the Pseudomonas control we use, as well as other environmental cosorts that we had. So what we ended up with was roughly 50 to 110 SNP differences per genome within the consortia. So between individual cells, there was this difference. Well, we're still interpreting this result. We do know it's going to raise some very interesting questions in experiments. Using the full-length 16S rRNA gene that we obtained from these genomes, we found that there are 5 distinct populations or groups of MMB existing in our site. So we built this phylogenetic tree to show their relatedness, and we compared them to other known and published on MMB existing across the world. Here you see A, B, and C, and the top of this tree have this morphology of a spherical shape with these sausage-shaped cells within the aggregate. And in the bottom of the tree, in D, E, and F, corresponding to these micrographs, you see that the MMB have an oblong shape to them and this unique H-shaped shell cells. So we then have the question, well, do we observe these unique morphologies in the samples from our site that are shown in these colored groups? And when we go to the fluorescent microscope, just using a DNA stain, we see, basically that the morphology is uniform across the different populations. So we needed to develop a method to look at the distinct groups or populations of MMB from our site and their morphology. So we developed a protocol for correlative fluorescent in situ hybridization, or FISH, scanning electron microscopy, shown here, where we would take a sample to a scanning electron microscope, obtain an image, and then use FISH to identify specific populations, here group 1 and group 4 from our sample site, and then map that back to the SEM image to look at their morphology. I'll discuss a little bit more of where this project is going in my last slide. We also wanted to look at the division of labor, and to do this, we use substrate analog probing, where you'll incubate the MMB in the presence of a methionine analog, or an amino acid analog that has the azide functional group. And so when new proteins are synthesized, these new proteins will have an azide functional group. They can then do azide alkyne click chemistry and covalently attach a fluorescent dye to the proteins. So what we hypothesized was that the MMB consortia would either not incorporate this AHA, and we'd see no fluorescent in the consortia, or every cell in the consortia would incorporate this methionine analog, the AHA, or alternatively, we'd see a division of labor where some cells are incorporating the AHA and newly synthesized protein and would be highly active, and other cells might not be making as much protein. We can also use other substrate analogs for peptidoglycan synthesis, and as well as fatty acid for membrane synthesis. We then use structured illumination microscopy, super resolution microscopy, to look at the incorporation or the synthesis of protein and peptidoglycan between cells within the consortia. And we did this with the peptidoglycan and the membrane and didn't observe any major differences. The dark spots you see here are just the light block from the polyhydroxybutyrate granules or energy storage granules within the cells. So this investigation is ongoing. To conclude, the current and future work of this project is to continue with the correlative microscopy, where we'll use stable isotope probing and link that with our scanning electron microscopy to get morphology and elemental composition, as well as Raman microspectroscopy to look at the chemical composition, and link that with FISH, and then finally use NanoSIMS to look at the localization of the CYP substrates in individual cells within the consortium to link taxonomy, physiology, and morphology. Of course, I couldn't do this without all my collaborators in my lab, as well as those at JGI and EMSL, and those that helped with the fieldwork. Thank you.